Okay. Okay. So, good morning, everyone, and welcome to this time's Zoom seminar. So it's really my great pleasure to welcome our today's speaker, Dr. Lisa Denzin from Rutgers University. So Lisa currently is associate professor in Children's Health Institute of New Jersey, Rutgers Johnson Medical School in Department of Pediatrics. So a very brief introduction about Lisa. So Lisa actually got her complete her undergraduate study in microbiology in University of Wisconsin, and then continued to pursue her PhD study also in microbiology in University of Illinois. And afterwards, Lisa actually decided to do her postdoc training in immunology in Yale University School of Medicine. So after Lisa completed her postdoc training, she started to her independent research program as assistant professor in Duke University Medical Center in Department of Immunology. And then she moved to Cornell in New York, also as assistant professor, where she got promoted as associate professor also in Cornell. From 2011, Lisa moved to Rutgers University as associate professor, and then to today, as I mentioned, that she working in the Children's Health Institute of New Jersey, the State University of New Jersey, and Rutgers University. So Lisa's work over the past 20 years or so has applied different techniques, including biomedical, cell biological, and immunological techniques to the question of how peptide loading of MHC class II molecules is modulated in vivo during immune responses. So Lisa's work has discovered numerous really groundbreaking discoveries, including the essential role of HLA-DM and the HLA-DO, which is H2o in mice, in modulating peptide loading of MHC class II, and have shown how these molecules work together to regulate immunity and perhaps autoimmunity. Lisa's lab has worked to understand the machinery by which H2O controls the immune responses to mouse retroviruses, and they show that H2O expression blocks the development of neutralizing antibody responses to retrovirus. They also defined naturally occurring alleles of human DOA and human DOB with alternate function, and then genetically link them to the outcomes of human chronic viral responses. So I think with a lot of spectacular discoveries, Lisa received numerous awards and honors, which including from 2001 to 2004, Breslow Scholar in Memorial Sloan Kettering Cancer Research Center. Also from 2001 to 2005, she got adult chair of junior faculty in also in Memorial Sloan Kettering Cancer Center. So I think today we are going to hear some recent work from Lisa's lab related to the non-classical MHC class II molecules. So we are very much looking forward to talk. Lisa, please go ahead. Thank you so much.
Thank you very much. Thank you, first of all, for the invitation. It's really nice to be able to give a seminar via this Immune Zoom conference to tell everybody a little bit about the work that my lab has been doing. And I thank the organizers for inviting me. It's really nice. And good morning to those of you on my time zone, and also hello to those of you across the world who may be getting ready for bed. So what I want to do today is actually tell you about the work my lab has been doing to understand the class II antigen processing and presentation pathway, and how the modulation of peptide loading really can influence immune responses. And I'm going to tell you right up front, I'm not going to give you a complete story. I really wish that I could, but what I really want to do today is introduce you to the pathway and the molecules that control this. it's an important pathway, but I think an understudied and we all think what we work on is underappreciated, but underappreciated pathway. And we have some interesting results that really suggest the pathway is much more complex than we would have thought. So I just want to start by, by just reminding everybody, it's so nice to talk to immunologists, by the way. I'm not going to give a big introduction, but I do want to remind people that the MHC, major histocompatibility, antigen processing and presentation pathways, have evolved really to surveil the intercellular compartments. So pathogens, bacteria, such as bacteria and viruses, parasites, etc., get into cells and try to hide from the immune system. And of course, what happens is proteins derived from these pathogens get broken down into small peptides, loaded onto MHC class 2 or class 1 molecules. And then the T cells come around and surveil the various cells in our body, looking for that MHC pathogen peptide. And when a T cell that's specific for that pathogen is recognizes an MHC peptide complex, the ultimate the response is the T cells get activated, they proliferate, and they go off and provide their effector functions. So just to remind you, the CD4 T cells are the helpers. So they really go out and orchestrate the immune response by secreting cytokines and telling the immune system what type of infection is present and to allow the development of the right response. And then CD8 T cells, of course, are cytotoxic T cells, and they surveil and go out and kill virally infected cells as a way, or bacterial infected cells, as a way to control the pathogen. And of course, this is all in the context of antibodies, which are important for that controlling the extracellular spaces. So my lab is really interested in this side of the equation here, and specifically how the peptides derived from these pathogenic proteins get loaded onto MHC class 2 molecules. So I'm going to, and the way we think about this is that presentation is really essential for immune system balance. And so during an immune response, you need just enough presentation of MHC molecules bound to pathogenic peptides to drive immunity. So to make the right immune response to the pathogen that has invaded the body. But not so much presentation, not presentation all the time, so that you provoke autoimmune responses. And so we think the balance of this pathway is essential. So the way the class 2 pathway works, and I'm just going to remind you of the steps in this pathway. This has a very well-studied pathway. We've known about the basics of this pathway for over 20 years, probably 25 years. And the way the pathway works is that the class 2 molecules, which are an alpha-beta heterodimer, assemble in the ER with another protein called the invariant chain. And the invariant chain is a specialized class 2 chaperone. It helps the class 2 molecules fold. So they fold together with the invariant chain. And the invariant chain has a couple important functions other than the chaperone properties. First of all, part of the invariant chain actually occupies the class 2 peptide binding groove in the ER, protecting it from premature peptide binding. Then once this complex is fully assembled, the invariant chain also donates a targeting signal, which is located in its cytoplasmic tail, that diverts, or that first of all allows the class 2 invariant chain complexes to move from the ER through the Golgi. And then this targeting signal diverts the class 2 to the cell surface. And then this class 2 invariant chain complex is endocytosed into endosomal lysosomal compartments. And of course, in these compartments, they're very rich in proteases, and a couple that are specific for the class 2 pathway, including cathepsin S and sometimes cathepsin L and a few other proteases. And it turns out that the invariant chain is a very elongated protein, is very protease sensitive. So what happens upon arrival in the endosomal lysosomal compartments is that the invariant chain quickly becomes cleaved away from the class 2 molecules by these resident proteases, leaving only CLIP in the binding groove of the class 2 molecule. And this CLIP peptide binds to the class 2 molecules with high enough affinity that in most cases it can't spontaneously dissociate from the class 2 molecules, creating an empty peptide binding groove. So there's another molecule called HLA-DM, which is over here in the ER. It assembles independently of the invariant chain. It's a class 2-like molecule. So in my model here, you can see it looks a lot like class 2, except it doesn't have a peptide binding groove. And DM traffics independently to these same endosomal lysosomal compartments via a tyrosine-based motif in the cytoplasmic tail of the beta chain. And what Dodm does is it actually associates with this class 2 CLIP complex via a lateral mechanism as shown here. And via this association, CLIP is popped out of the groove of the class 2 molecule. Dodm then holds on to this empty class 2 molecule, preserving what's thought to be a peptide receptive state. So it's chaperoning the class 2 molecule and prolonging its half-life in this endosomal lysosomal compartments until antigens that have been internalized into these same compartments or antigens that reside in these compartments can be broken down by the same resonant proteases into small peptides that are 12 to 24 amino acids long. Eventually, a peptide will bind that, of course, has the right binding motif that can bind to a given MHC molecule. It binds to the class 2, Dodm is released, and then Dodm does something that is probably its most important function, and that's called Dodm-mediated peptide editing. And what is meant by this is that Dodm is checking those class 2 peptide complexes to make sure that the peptide that has been loaded has a very slow off rate or overall high affinity. And this ensures that the class 2 peptide complexes that arrive at the cell surface have very long-lived peptides bound. Otherwise, there would just simply be exchange at the cell surface and you'd override this intercellular compartment. And it's thought that if the peptide is not high affinity or doesn't have a slow off rate, that Dodm will actually kick it out of the groove, allowing for another round of loading with a different peptide, which is then checked again. And once this peptide can no longer be removed by this association with Dodm, it moves to the cell surface for activation and propagation of CD4 T cell responses. So this is, as I've already said, a pathway that's been studied for a very long period of time. We've known about Dodm since, I guess, the mid-1990s. And later in the 1990s, it was discovered that another molecule called HLA-DO in humans, or H2o in mice, associated with Dodm. And this was discovered by Lars Carlson's lab, I think in 1997 or so. And what DO is it also, like Dodm, is a MHC-like class 2 molecule. It's actually more similar to classical class 2 than Dodm is. So all these molecules are structurally related, but DO and class 2 are much more similar. Unlike the class 2 molecules, which of course are highly polymorphic so that they can bind all the peptides ever generated as a surveillance mechanism to clear pathogens, the DO molecule is the alpha and beta chains of the DO molecule are basically non-polymorphic, and I'll come back to that later in my talk. And what Lars's lab showed a long time ago now was that DO associates with Dodm in the ER. So this complex is formed very early in the biosynthetic pathway, and it's transported as a complex to late endosomal lysosomal compartments where the complexes basically reside in cells. And this is a recent confocal image taken by Nelson Cole, who's in the Paul Roche's lab at the National Cancer Institute. And he's taken mouse dendritic cells here and fixed and permeabilized them and stained them with antibodies for H2M, so the mouse version of Dodm, and H2o. And all I really want you to see here is that DO and Dodm pretty much perfectly co-localized. So H2o binds to H2M, gets pulled into these endosomal compartments where H2o lives. And so I was still a postdoc at Yale in Pete Cresswell's lab. Lars had published his paper, and Pete and I had recently worked on Dodm function, and we said, wow, anything that's that tightly bound to Dodm is tightly bound to Dodm is likely to alter its function. And so we actually went on to show, and I did put the data in here, that DO actually inhibits Dodm. And many years later, actually in 2013, so about 10 years ago now, the crystal structure of DO bound to Dodm was finally crystallized. I'm going to start up here, though, with the Dodm class 2 crystal structure, which was also published around the same time, actually a year earlier. And what this crystal structure showed is that Dodm and class 2 associate together via this lateral mechanism. And I'm not going to talk about how Dodm actually plays with the peptide binding groove and allows peptide to bind, but that's the basic mechanism by which it's thought to happen. But for this talk, what's important here is that DO and Dodm in the crystal structure bind in a very similar manner to each other. And you don't have to be a crystallographer to look at DO and class 2 here to see that they look nearly identical. So what this set up was the idea is that DO is simply a substrate mimic of MHC class 2, and it's a competitive inhibitor. So DO binds to DM, preventing DM from interacting with class 2. And the result is that you have an inhibition of DM-mediated peptide loading of MHC class 2 molecules. So to put this into the model of how we think this happens is that, so now in our model here, we have the DM-DO complex. And just to remind you, this complex is formed in the ER. If you express DO in the absence of DM, DO never leaves the ER and it's quickly shuttled out to the cytosol and degraded. So it's an unhappy protein without DM. So this complex forms, and via this targeting signal in the beta chain of DM, the complex is diverted or localized to these endosomal compartments, as I showed you in the last, in a couple slides ago, I guess, in the immunofluorescence. So basically, because DO is now bound to DM, this prevents DM from interacting with this MHC class 2 clip complex and keeping clip out of the groove. And what happens then in cells where you express DO is you have an increased abundance of class 2 clip at the cell surface and decreased class 2 peptide. So this sets up the or shows that DO inhibits DM-mediated peptide editing and clip removal, and also inhibits this editing that DM does to shape the peptide repertoire. However, in all primary antigen presenting cells that we have looked at, so mainly DO is mainly expressed, forgot to mention this, in dendritic cells and B cells. We don't see much expression in macrophages. So it really seems to be localized to these two subtypes of class 2 positive antigen presenting cells. So in primary DCs from humans and mice and B cells from human and mice, what you find is that about half of the DM is actually bound to DO, and there's always a free pool of DM. So what is really happening in the endosomes of cells is that some of the DM is inhibited, and you do see an increase of clip at the cell surface. However, DM is still there, it's active, and peptide loading does occur. So really what happens is that peptide loading is controlled by DM, but modulated by DO. And in general, it's a down regulation of peptide loading, but it turns out it's a little bit, as biology always is, it's a little more complex than that. So more recent mass spec-based studies have really gone in and asked the question, what is the impact of having this DM-DO complex, again, together with about half the DM being free, on peptide loading? Does it change the repertoire of peptides that are actually presented at the cell surface? And the answer is yes, but not really as significantly as originally thought. I don't have the data on here, but overall, it's about 10% of the peptide repertoire that changes in the presence of DM, in the presence of DO. And what you see is, first of all, there's absolutely an increase in MHC class 2 clip. So if DM can't interact with that MHC clip intermediate in the pathway, clip doesn't come out of the groove and instead moves to the cell surface. But after that, you see a really mixed change in the peptide repertoire. Some peptides are actually enhanced by the presence of DO, and these are likely peptides that would normally be edited out of the groove of the class 2 molecule by DM if you have full activity of DM. You actually find some peptides that are very poorly expressed, and these peptides tend to be enhanced in the presence of DM. You have a lot of peptides, most of the peptides that actually don't seem to care at all. So they're there in really equal abundance, as best as one can measure these things using mass spec-based approaches. I think that's an important caveat. And then the only difference that's really apparent is that in the presence of DO, you have a set of peptides that aren't present when you have only DM there. And it's thought that these are the peptides that are really susceptible to DM-mediated peptide loading. So these would be peptides with fast off rates and overall fairly low affinity. So having DO there not only inhibits peptide loading because you have more class 2 clip at the cell surface, but it's really thought to broaden the peptide repertoire, allowing lower affinity peptides to be bound and presented. And the field is really thinking about this as, in the presence of DO, you have this broad, diverse, lower affinity relatively lower affinity set of peptides presented. And I'll come back to this, but maybe this is important for tolerance. It's allowing a representation of self-peptides at the cell surface that drive peripheral and maybe even central tolerant functions. And so maybe this is what does promote T cell tolerance. On this other hand, when you have only DM, you have a more restricted, higher affinity set of peptides. Perhaps the immunodominant peptides are enriched. I think this remains to be proven. And the idea is this would be a peptide repertoire that would promote an immune response to an offending pathogen. So what happens upon activation of antigen presenting cells? Well, it turns out that if you take a dendritic cell and activate it with TLR ligands or via other mechanisms, what happens is DO is actually downregulated very rapidly. It happens within hours, actually. And it's downregulated much more so in the CDC2 subset of dendritic cells. So these are the dendritic cells that are thought to be more specialized for class 2 presentation, and less so in the DC1 so-called tolerogenic DC population. DO is also massively downregulated in germinal center B cells. So germinal center B cells basically express little to no DO protein. And then, of course, you're going to have this situation where you have only DM and other DO negative antigen presenting cells, which we think are most of the macrophages in vivo. So what my lab has really been interested in since I left Pete Creswell's lab at Yale is trying to understand what the ramifications of DO modulation of this peptide repertoire are on the immune response in vivo in mice. And we really think, again, that this is a balancing act and that DM and DO are really essential for maintaining this idea of having just enough or just the right presentation at the right time, but not too much presentation potentially to prevent against autoimmune responses. So the H2o knockout mice were made in the late 90s by Lars Carlson's lab, actually, and we quickly got them. And I will say that they have a very wimpy phenotype. We've worked on them extensively, and I'm not going to present a lot of negative data in studies that we've done over the years to try to figure out what O is really doing to modulate, what the ramifications of O are on the immune response. One thing we were able to show using a complicated cell transfer system and competition assays is that having H2O expression, so in wild type cells, really restricted the access of B cells into the germinal center. So it was dampening antibody responses. And in this paper, which was published a long time ago now, what we showed is that there was definitely a change in the peptide repertoire that changed the T cell responses. And our data really suggested that the change in peptide presentation was actually relatively subtle, like maybe twofold at most. But yet it was enough to really dictate how many of the activated B cells in the periphery could go into the germinal center and ultimately contribute to the antibody response. So this fits very nicely with H2O being a negative regulator of the immune system and having some control over the B cell response. And I think the important question here that I'm going to admit right up front that I'm not going to answer is, why does the immune system want to dampen these responses, and why do you need this negative regulator of the immune system? In this case, we have not gone on to test this. I need to come back to this idea, but what we think maybe is going on here is that the immune system doesn't always want to respond to a pathogen or an antigen unless there's enough of it around. So it's setting a threshold to make it a little bit harder to make a full-blown, isotype switched antibody response to an antigen when you don't have a lot of antigen around and maybe other pathways in the immune system can clear it. But that's something we need to come back to and really test formally. So what I want to tell you about today is that my lab has, as I've already said, spent a lot of time trying to figure out what this molecule does, and we have a lot of negative data. And I learned that this is actually a seminar series that's really for trainees, and I think it's important to say that it's important to stick with things, and sometimes you need a little bit of luck, and that's what happened to us. So I want to tell you a little bit about this retrovirus story and immunity part of this balancing equation. And so H2o mouse retroviruses, I think that most of us know that mouse mammary tumor virus is an endogenous chronic mouse gamma retrovirus. So this is a nice model for looking at the retrovirus that actually infects the proper host, the mouse, as opposed to trying forcing human retrovirus such as HIV to infect mice, which never works very well. What this does is it actually takes a virus that actually infects mice and allows you to look at the immune response in a natural setting. And so this is a chronic infection in mice, and it's a chronic infection in laboratory strains of mice because these laboratory strains of mice get infected and they're susceptible to MMTV. They never make an immune response to it. So in your mouse room, in your colony, your mice have MMTV. They get they're infected. And what happens is the virus ultimately traffics to the mammary gland. And when female mice have babies and they nurse the babies, the dams nurse the pups, the virus is transferred via the breast milk to the babies. And basically, this is a cycle that continues on and on and on. And there is a ramification of this infection, and I can probably figure out what this is. If mice get to be about 1.5 to 2 years old, some of them will develop tumors from this mouse mammary virus infection. So in the wild, this doesn't really matter because wild mice don't live that long. And in the lab, we tend to not keep our mice around that long. And so we don't really see the negative impact of this viral infection, and most of us ignore it. Most of us, except for my collaborator, Tatiana Glinka at the University of Chicago, who has spent her entire career working on these mouse retroviruses. And what she did is when she first started her lab a long time ago, she moved to the Jackson's lab and she had access to every strain of mice ever available. And she wanted to ask a question. Well, most laboratory strains of mice are susceptible to MMTV. Can I find strains of mice that are actually resistant? And the answer to this very long story, the short answer is that yes, absolutely. She found several, but one of these, and I'm going to talk only about this one today, is IL and J mice. She found that they're resistant to MMTV. This is actually what they look like. They're very cute, much cuter than the normal laboratory strains of mice. And what her data showed, which is down here and has been published on extensively, they become infected with MMTV. They develop neutralizing antibodies. So unlike the susceptible mice that pretty much just ignore the virus and never make much of an immune response at all to it, these mice make a strong antibody response. So she is a mouse geneticist as well as a retroviral biologist. And so she went gene hunting and using a series of positional cloning, congenic mouse strains, phenotype rescue experiments, and ultimately whole genome sequencing, which took her 17 years. And I think that's an important message for people that, if you have a question that's important, you stick with it and you can follow through and ultimately, come to an answer. So 17 years later, the whole genome sequencing data was coming off. And it turns out that Tatiana's husband postdoced with my husband. And so we sort of knew each other. And as the data was coming off, she had some really strong evidence from the whole genome sequencing that the main gene that mediates this phenotype, which she had called viral infectivity controller 1, was actually the beta chain of H2o beta. So this negative modulator of the immune response. So she contacted me and we set up a collaboration to actually test this idea. So if H2OB was VIC1, then mice lacking H2O should actually make this neutralizing antibody response. So she grew up virus and sent it to me and I infected. We had H2OA knockout mice at that point, but this H2O is an obligate heterodimer. So if you knock out the alpha chain, there is the molecule number assembles and there is no H2O. So we infected the OA knockout mice, wild type mice, and she infected some control mice at Chicago with the same virus. And we bled the mice 3 months later and asked what happened, do we have any antibodies? So this is the ELISA Tatiana did 3 months later. And what you can see is her cohort of positive control mice, which were just one of her congenic strains, made antibodies that were specific for MMTV. The wild type mice in my colony did not, but the knockout mice actually did. So this was great. Of course, the hallmark of this response is neutralizing antibody response. Antibodies don't mean much unless they actually neutralize in this infection. And so she took the sera from these mice and added them to retrovirus and then put them into a highly susceptible host, which is BALB/c mice, and then read out MMT infection to ask, was the virus neutralized? And the answer is, so first of all, her control mice neutralized very well. The wild type mice in my colony very poorly neutralized the virus, and the knockout mice nicely neutralized the virus almost as effectively as her congenic mouse strain that was highly efficient at neutralizing. So this really sets up the idea that OB is actually VIC1, and that we had uncovered a mechanism that was controlling the neutralizing antibody response. I have to say her genetics showed that this is a recessive gene VIC1 is, which fits with the idea that what she's looking for is a negative regulator of the immune system. We then we knocked OB out just to make sure that we were right, did the same experiment, infected those mice with MMTV. And again, the knockout mice, but not wild type made MMT specific antibodies. And this antibody in the knockout neutralized the virus, but not in wild type mice. So this really nicely shows that OB is VIC1, and that under normal circumstances in those susceptible strains of mice, H2O is blocking the development of neutralizing antibody responses. So we next turned to try to figure out, well, what's going on with IL-NJ-OB? What's wrong with it? Is it not there? Is there no protein? So the whole genome sequencing and then a lot of other sequencing showed that there's 4 unique mutations in IL-NJ-OB compared to O-beta from the MMT susceptible mice. And these are shown on the crystal structure of DM and DO. And 3 of these mutations were in the immunoglobulin domain, so the bottom part of the molecule. And it looks like they're really at the interface of DO, DM. It turns out that they're really not pointing towards DM, they're more pointing away from DM. And then there was a charge change in the cytoplasmic tail of DO beta. And I'm going to tell you right now, I thought for sure this is absolutely the problem. And because cytoplasmic tails alter targeting of molecules, and perhaps what was going on is that there's mistargeting of IL-NJ-O beta. But before we asked that question, we basically said, well, is there protein at all? So first of all, IL-NJ mRNA levels, OB mRNA levels were normal. And so we thought, well, what about the protein? Maybe there's no H2O and this is simply a knockout mouse. And that's why they're making neutralizing antibodies. So, whoops, that doesn't belong there, sorry. So what we did is we just purified B cells and we ran Western blots and quickly able to show that compared to susceptible strains, IL-NJ-OB was expressed at normal levels, M was there at normal levels, and here's acting as a control. There is a slight shift in the molecular weight of IL-NJ-OB. I actually still work in the lab, so I actually took on the mass spec sequencing of this. There's absolutely no deletions, et cetera, in this protein at all. And a series of very long and painful experiments basically showed that this protein migrates faster on SDS page gels just because of that lysine residue in the cytoplasmic tail, which causes the protein to bind more SDS. So it's simply just a change in mobility on the SDS page gel due to that, the ability to bind more SDS. So with knowing now that we had normal levels of protein, we next thought, well, okay, maybe these mutations in the immunoglobulin domain were disrupting the DO-DM interaction. Remember, DO is a competitive inhibitor. So if DO wasn't bound as tight to DM, perhaps what's happening is it moves into endosomes, the complexes dissociate, leaving free DM, and you get very efficient peptide loading, as shown in that slide of the peptide repertoires. So to test this, we did immunoprecipitations to probe the interaction between IL-NJ-OB and H2M. So using an antibody specific for H2M, we immunoprecipitated H2M from B6 mice, wild type mice, and IL-NJ mice. And so you see here's the MA protein. We also did it from OB knockout mice, which have M, but no O. And what you can see is that in B6 mice, we efficiently precipitate H2O with H2M as would be expected. And we see the same result in IL-NJ mice. Again, here's the shift due to excess SDS binding to IL-NJ. So what this tells us is that at least biochemically using this assay, H2O seems to bind normally to H2M in island J mice. And it does not fit with the dogma, which would suggest that if this protein is non-functional, it should not be bound to H2M. Also, we've now done immunofluorescence and EndoH experiments that have proven that H2O from island J mice, this mutant H2O, will actually traffic and reside in endosomal compartments. Everything looks perfectly normal. We don't see any differences. So this is a bit of a head scratcher for us. It's one of the first of many that I'm going to share with you. So what we next did was we're like, well, is it really a null mutant? We assume it is, but based upon the phenotype of making antibodies, which matches the OB knockout and then OA knockout, but is it actually a null allele? And so to do this, we had to turn to mouse genetics to do a couple tricks, because the only antibody we have for measuring class 2 CLIP. So the first way we wanted to do this was to ask, well, if O is active, it should inhibit H2M, resulting in higher levels of class 2 CLIP in the mice if it's active. So what we did is we took B6 mice and we crossed them with island J mice to allow us to have the right class 2 molecule present, because the only antibody to measure class 2 CLIP is specific for IA of B, so B6 class 2 CLIP complexes. And island J mouse are HLA or are J haplotypes, so you can't use that antibody in these mice. So we created F1 mice, which allowed us to have an A of B molecule there and a wild type version of OB. And then we also similarly crossed island J mice with the OB knockout mice. So in this situation, you have one copy of island J, so this allele we want to assay, compared to with no wild type, comparing it to wild type versus this one that we want to test. And so basically this is an island J OB het that only expresses island J OB. And when we measured CLIP to read out H2O function, what we found is that this is now FACS analysis of B cells looking at the I of B CLIP levels. And this is the level we see in wild type mice. And when we looked at the OB that had one copy of island J mice compared to the het here, we saw a reduced level of class 2 CLIP complexes that was highly significantly reduced. And this is the shift we always see. It's about a 20% change of class 2 CLIP in OB knockouts. And that's actually shown here. So if you now stay in OB knockouts compared to wild type, there's about a 20% decrease in class 2 CLIP. It's highly, highly, highly reproducible. So we 100% believe it, even though it is rather tiny or rather small. Importantly here, what we show is that the heterozygous OB mice actually do have wild type levels of H2O. And so that's important because here we have a heterozygous B6 mouse that has wild type levels. And when we get rid of that wild type allele, we now shift down to the knockout level, supporting that this island J OB is a null allele. So what I've shown you so far is that there's these 4 unique mutations in O beta compared to the MMT susceptible mice. The levels are normal. It surprisingly interacts normally with H2M. However, it's still a null allele because A of B CLIP levels are similar to OB knockout mice. And I'm not going to, just in sake of time, I'm not going to show you some hybridoma data that we had that also supported that it's a null allele. And I have to say, this is a real head scratcher for us because it wasn't what we, it didn't fit the dogma of the way the pathway is supposed to work. H2O is supposed to be a competitive inhibitor of H2M mediated peptide loading of class 2 molecules, but how could it not inhibit? It's bound, but yet it wasn't inhibiting. So just for what we think is going on for the immune response in island J mice, which is ultimately the question we want to address, is that in retroviral susceptible strains, what we think what we know happens is that the B cells get infected, they take up the virus, and we think that in the presence of wild type H2O, that presentation has been dampened, leading to no development of a CD4 T cell response, and then ultimately no isotype switched antibody response that neutralizes the virus. However, in the retroviral resistant strains, so either island J mice or knockout mice, You have slightly better presentation based upon our previous results in the knockout mice that I told you about. You get slightly better presentation, and that is just enough to drive the CD4 T cell response, leading to the neutralizing antibody response. I have to say this part in here is really a black box. It turns out there's no reagents to let us test this in MMTV mice. There's no peptides, there's no T cells. We've tried extensively to create those reagents and have yet to be successful. So it's something we're working on, but this is the idea of what we think H2o is doing. So with that in mind, I want to switch quickly to this idea, well, okay, this happens in mice in retrovirus infection. But what about humans? Does DO have a similar role in humans? Is it biologically relevant to the immune response to viruses? And to do this, we first had to look for variants of DO in human that altered function. And I've already mentioned that DOA and DOB are non-polymorphic. The reality of it is that they're mostly non-polymorphic. There are rare variants that are present in humans. And so what we did was Ali Khan, who's now at the University of Chicago, searched the exact database. So that's the 60,000 exomes database, together with the 1,000 genomes database, just looking for variants of DOA and DOB, coding variants that might alter function. And what Ali found was that for DOA, he found 107 variants, and for DOB, 94. There's also some common alleles for these. Then these are alleles that are more highly represented in nature. And I have to say for DOA, one allele accounts for 99% of people, of individuals. For DOB, there's two or three alleles that pretty much account for 99%. Found several frame shift stop mutants. So these are ones that would probably make no protein at all. And then 95 and 79 missense mutations. And then for DOB, there were a couple splice mutants. And so what we decided to do was to take a selected series of variants that were all along the coding sequences for both DOA and DOB. And we set up an in vitro assay that allowed us to assay DO function. And this was, I don't have the slide, but basically it's a transfection assay where we put the DO variants into HeLa cells that express class 2 and DM, but not DO, and then measured class 2 CLIP levels. So DO inhibits DM, resulting in higher levels of CLIP. So we measured CLIP for all these variants, as well as DO protein levels, DM protein levels, you name it, we measured it. And then read it out for changing function. And I'm only going to show you the summary of this data. And as I said, we took DOB and DOA variants that were located all throughout the coding regions. And we did this on purpose because we knew from looking at the IL-J data that those mutations were not obvious. There was nothing that said, this is going to be a mutation that's going to change function. So we wanted to really lay a very broad network across the proteins to make sure we didn't miss something. And Francesco Virtus and Austin Graves in my labs, a postdoc and a graduate student, did these assays. And what they found was that for DOB, and this was Francesca's work, 14 of the 32 variants that we assayed, including the common alleles, had altered function. And I don't really want to go through this because it's been published and it's kind of boring to go through mutant by mutant. But the basic idea is that we found some that were null. So these frame shift mutants made no proteins and did not inhibit, obviously. We found a whole bunch of proteins for DOB that actually had reduced function. And then surprisingly, we found a couple that actually had increased function. They actually inhibited DM better than the wild type, meaning the most highly represented alleles of DO and A and DOB inhibited. For DO, we found slightly different thing, but again, 50% of what we assayed actually had altered function. In this case, we found a lot of alleles that actually had increased function. And DOA is the part of the DO protein that actually directly interacts with DM and makes the most contact. So it appears that most of these mutations are perhaps making that binding interaction tighter. However, we did find a couple null alleles and then also one that actually had decreased function, as we found for DOB. So importantly, however, we did find two variants that had the IL-J phenotype, meaning they made wild type levels of DO protein. that bound to DM and was still tightly bound using that immunoprecipitation assays, but yet did not inhibit DM-mediated peptide loading. So here is another example of something we didn't understand. How can this be happening? Also found not just for the mouse protein, but for the human protein. So the real reason we did this assay, however, was to look for variants that ultimately we could link to human viral infections. And of course, we're very interested in HIV. I'm sure everybody has already made the link that, do these DO variants potentially play a role in the immune response to HIV? And there's a group of individuals that get infected with HIV called the elite controllers or the super controllers that really control the virus and often aren't even discovered to have HIV until they're screened for other reasons. And they're like, wow, I'm either HIV positive, but yet they're controlling it. And a subset of these individuals actually make a strong neutralizing antibody response. So we thought, wow, maybe some of these variants are expressed in these individuals that make these antibody responses. This is where biology was hard on us, though, because to do this type of assay, we needed SNPs in these that marked these variants that would then allow us to go parse the databases to test this. So we were unfortunate / fortunate that we found three SNPs in DO that map to several of these mutants, and then 2 in DOA that mapped. And so initially we were focused just on DOB because it was the OB story, and Tatiana and I contacted Bruce Walker's lab, who has the largest cohort of elite controllers in the world, and gave him the SNPs and asked, can you link this to the antibody response? And the answer was, unfortunately, no. There's lots of problems with this type of analysis. Clearly, we're restricting it only to a couple alleles, and the cohort's really small, and these alleles are very rare. So we don't know at this time if there's a link or not. Our data didn't show one, but that doesn't mean that there isn't one. And this type of analysis is really thwarted by the fact, or really hurt by the fact that assembly of the human MHC is still nearly impossible from sequencing data. And so once that becomes more possible, which I don't do genomics, and this is not far from my area of expertise, but my understanding is once the reads get longer and longer, it'll become much, much more possible to assemble the whole MHC region, which will then allow us to then query these databases in a much more logical manner. So with that disappointing result, we decided to just take a different approach and say, okay, not HIV, but what about other chronic viral infections, and specifically chronic viral infections for which antibody responses are important. So it turns out that human hepatitis B and C virus clearance has been shown to be linked to the production of an early and broadly neutralizing antibody responses. So in the case of hepatitis C, virus infection, about 20% of individuals make this antibody response and clear the virus on their own. And for human hepatitis B virus, it's about, I think it's 90%, so much more efficient. But the point here is that we have a viral infections that a neutralizing antibody response can clear it. And this gave us another opportunity to ask the question, are any of these variants linked to these responses? And so to do this, we had to take a little bit of a trick, and we took advantage of the fact that previous GWAS studies, shown here in this case, linked hepatitis C viral persistence, so this would be no antibodies and the virus wins, to one of the class 2 alleles, and in this case, it was DQA2 and a certain allele of it. So here's the human MHC on chromosome 6. And what I want you to see is that here's HLA DQA2, which is actually very close to DOB. And so what we did is we took this SNP and asked if any of our SNPs for which we had actually a SNP, or any of our mutants for which we had a SNP, were in linkage equilibrium. So were they linked very tightly to this DQ2 allele? And actually we found one that was, and it's this DOB G77V allele, which was completely in linkage disequilibrium, so 1. And what that means is that you can't distinguish if DOB or this allele DOB or HLA-D, this allele of HLA-DQA2 is linked to hep C persistence. It could be either of them. And what was really cool about this linkage, and exciting, I guess, is that this was one of our super DOs. So the DO function was increased. So it was going to inhibit DM more, which would be associated, presumably, and I use the word presumably because we don't know this, with the decreased antibody response. And this fit very nicely with the fact that it's associated with hepatitis viral C persistence. So one link like this, and I think you're lucky. And I mean, I think, you know, maybe it's just luck and it's not real. However, with our DOA mutants, we were actually able to link to hepatitis B persistence and action clearance in this case. And again, we took advantage of the fact that HLA is right next to DOA, and previous GWAS studies had shown that there is a SNP in DPA1, so the alpha chain of DP, that was very close to DOA, and that one allele of this was linked to persistence, so no antibodies, and the other allele was linked to clearance. So when we looked at our DOA SNPs, what we found is that one of them was in linkage disequilibrium with DPA1, and this one was an allele that had, again, this super DO phenotype, so more DO, presumably less antibody, and this allele was linked to persistence. And then another allele was linked to this same SNP. And this allele is a very strange one. We don't understand the biochemistry of this. It's a phenylalanine at 114 that's changed to a leucine, but actually gives us no DO protein at all. So there must be some problem with assembly or something, and it's getting degraded. But this is basically a null phenotype then, which would lead to antibodies being made and was linked to the hepatitis B clearance. So this is really correlative data that suggests these variants, although rare, may be helping to control the outcome of these chronic viral infections in humans. So, oh, and I also want to say that there has been sort of a smattering of papers that have suggested links to DOA and DOB with HCV and HBV. And this is work done mainly by several Chinese groups that just have these beautiful cohorts of I guess I shouldn't call them beautiful cohorts because these people have hepatitis B and C, and that's not a good thing. But they have just a really powerful pools of samples with really, really strong data analysis that have also linked, in this case, this is a link with DOB to Hep C. So we actually think that this we might be onto something, but it's going to take, as I said, longer reads of the MHC to be able to put this together. So I wanted to come back to this H2o mice, and I think this is a good story for students to hear and how persistence is really important. So what we next wanted to do, since we were unable to make T cells and do the actual immunology experiments that I wanted to do, we decided to come back to this mutant and try to figure out which of these amino acid changes were mediating the null phenotype, with the idea we could use that then as a tool to work backwards in the immune response. And so what we did was we decided, and this is just the stick model showing the 3 changes in the immunoglobulin domain and the 1 change in the cytoplasmic tail. There is another change here. I don't want you to get confused, or 2 changes. These changes are unlikely to be important because they're present in all of the susceptible strains. So we don't think they mediate function. And it's just these 4 amino acids in red here that are likely causing the phenotype. And so what we wanted to do was to make the knock-in mutations one at a time and ask what is mediating this function. And I thought for sure it was absolutely this charge change in the cytoplasmic tail. The crystallographers and structural biologists told us it was going to be this one because this was in a portion of the molecule where this leucine, which is highly conserved in all of the DOs throughout evolution, change to histidine was going to push the kink the chain out, push towards DM, and was absolutely going to be the thing that was mediating the function. So we created all these individual knock-in mice. So onto the B6 background, we knocked the N into where the S is, and I into V, and L into H into L. We also made a double mutant where we had the N and the I together on the B6 background, and then we made the tail mutations. And I'm going to go through this data really quickly because it's negative. It wasn't any of these mutations in isolation or these 2 combinations. So here is the CLIP levels, class 2 CLIP levels in the knockout, so about 80% of wild type. And then if you look at any of these single and double knock-in mutations, CLIP levels are perfectly normal. If you infect the mice with MMTB and do neutralization assay, they very poorly neutralized compared to wild type. So in isolation, it wasn't any of these, it wasn't simple, right? It was going to be hard. So we kind of scratched our head and went, all right, how do we do this in a logical manner that allows us to not make 1 million mice? That would take forever. And so what we did is we turned to bioinformatics, and we discovered, actually Emily Cullen, was an MD PhD student in Tatiana's lab, discovered that there's a wild type strain of mice, Castaneous mice, that actually have these 3 Ig mutations encoded in the mouse, but they have a wild type tail. And again, I forgot to point out that I was completely wrong. The single mutation of the change of the glutamic acid to lysine, no difference at all. So the idea that my idea was not right at all. So I'm often wrong, but I think it's important to, have an idea, and then when you're proven wrong, to admit that you're wrong. So I was wrong. But what's cool about this is because we had these Castaneous mice have these 3 mutations all together, this let us ask the question fairly easily, is it the 3 NIH, and how perfect is that, right? I'm in an NIH seminar telling you about NIH mutations. So we took Castaneous mice and infected them with MMTV and asked, do they make antibodies? And actually they do. They made as many, if not more, antibodies as IL-J mice. And here's a negative control. And these antibodies very efficiently neutralize the mice. So we took advantage of the F1 assay that I told you about before, where we crossed Castaneous mice with either the OB knockout or with B6 mice, and then asked what happens to class 2 CLIP. And what we found is, first of all, here's our wild type levels in our control mice. And when we have only the Castaneous allele of OB present, we have the lower levels of OB. So this said, this is likely a mutant allele. So they have low class 2 and they express antibodies. So that was really promising. So we did the next obvious experiment, and that's to create the knock-in of these 3 immunoglobulin domain mutations onto the B6 background. So here we have our wild type SBL and our knockout NIH. And when we assayed these mice, we were happy. So here's the class 2 CLIP levels. So here's our wild type controls in this experiment. And here's the NIH mice. So they are lower. They're not as low as the knockout. And when we assayed for antibodies, the wild type make a little bit of antibody, NIH make significantly more antibodies overall, although again, the level is not quite as high as you see in a knockout mice. So we have recapitulated the phenotype of the IL-J mice, and we do believe that these NIH residues really mediate this effect. However, I will tell you that we have now taken the NIH mice and are in the process of knocking in these tail mutations to see if we can bring it down to, this 80% level, and is the tail also playing a role. So in the meantime, though, we now have created finally the perfect reagents to let us do the proper biochemistry. I skipped over some of the caveats of some of the other biochemical analyses we did to look at the interaction of O and M because of antibody problems. Basically, these mutations create problems, and in particular, the mutations in the tail actually altered antibody binding. So now we finally had a wild type OB expressed in vivo and a mutant NIH, as I'm going to call it, OB, also in B cells, that we could use antibodies to the cytoplasmic tail of OB, pull the complex down and ask, is that interaction really as robust as our preliminary data in other assays had told us? And this is an experiment done recently by Sarth Shaw, who is a technician in my lab. So he took B cells, purified B cells from B6J mice, so wild type mice, and B6J NIH mice, and just simply started by blotting the lysates. So here's a beta actin control, and then 3 B6J and 3 B6 NIH mice, and you can see OB levels are similar in both of them, and also our H2M levthat So he then did the immunoprecipitation assay to ask, what about that interaction? Is it that IL-J, I'm sorry, not that NIH OB doesn't interact with H2M as well as we think it does, as in our old data, it just wasn't done as well as it could be. And what Sarth found is that when he immunoprecipitated using an antibody to the cytoplasmic tail, so these polymorphisms in the proteins cannot be interfering with the IP. What he found is that OB pulled down, H2O IP pulled down the same amounts of O beta, similar levels of M beta. And when he did the converse IP, he had now used an antibody to the luminal domain of H2M to pull down the complex, and then blotted for the OB cytoplasmic tail. Again, the polymorphisms aren't going to play a role here. He found similar levels of O bound to M. So in both cases, using the reciprocal IPs, we see absolutely no evidence of disruption of this MO interaction, which is not what we would expect, given that this molecule doesn't inhibit DM. So I'm going to bring my guy back here going, what's going on? I'm going to be honest, I don't know what's going on, and we're now in the process of using these optimal reagents and doing mass spec to look for proteins that preferentially interact either with wild type or NIH that might mediate some sort of functional effect that we don't understand, or some mechanism that we don't understand. We actually do have a protein that preferentially interacts with wild type and not NIH, but I don't believe it yet, so I'm not willing to stake my reputation and tell you about it yet. And we have a slew of other proteins that look like they may be involved. So we're in the process of setting up proximity labeling, et cetera, to really further analyze this. And I'm really excited to be able to do this. And as I've already mentioned, we are making the knock-in with the tail mutations just to make sure that it's not a combination of all of these mutations together. So then I just want to quickly share another story with you that highlights the idea that although these pathways are very well studied and everybody thinks we know how it works, we don't. And this is a story that is really good for students to hear. And we've actually discovered other modifiers on the MHC pathway. And so we all use mice, B6 mice. A lot of us get them from Jackson, so the B6J strain of mice. And this has been at Jackson Labs since 1948. But in 1951, they shipped some of these mice to the NIH, and the NIH developed their own strain of B6 mice, which is commonly referred to as B6N for NIH. And then the NIH sent them to Taconic, and they have a version of B6N Taconic that was developed in 1991. So there's these strains of mice. And for reasons that I'm not going to take the time to tell you about, we accidentally discovered that B6N mice, both from Taconic or from the NIH, actually have this low CLIP phenotype. So these are MHC CLIP levels. Here's B6J set to 100%, and here's this 80% drop of CLIP levels that looks like an H2o knockout. So this was a bit of a head scratcher for us. So we went ahead and infected the mice, B6N mice, and you see they make antibodies compared to B6J that efficiently neutralize the virus. So you've been listening to me talk for a long time now, and you probably are going, wow, that's the H2o knockout phenotype. What's going on here? Well, it turns out that there's no polymorphisms in the coding regions for H2M, class 2, H2O, the invariant chain of the cathepsins that are important for this pathway. So we thought, initially we thought maybe something had happened to H2O in the M strain, but no, not at all. We biochemically confirmed that all of these levels are identical in B6J and B6N. So again, here we go. Well, what's going on? We thought we knew everything about this pathway, but clearly we don't. So Tatiana's a geneticist. She said, I'm going to just see how many genes there are. This could, 2 generations and we'll have an idea of what's going on here. So she did the genetic crosses by taking B6J and crossing with B6N and then asking what are the CLIP levels in the F1 generation. And they remained high. So this tells us that the genes expressed in the B6J strain of mice are the dominant alleles. She then did now I'm going to go fast because I know I'm going long here, but she did the appropriate back cross, which is you back cross the F1 with the recessive strain of mice. You create N2 mice, and then you ask what's going on with CLIP. And what she found is that they really segregated into 2 groups. So you had some N2s that were CLIP high and some N2 mice that were CLIP low. And doing the math and following classical genetics, what this tells us is that 25% of the mice were CLIP high and 75% were CLIP low. And what this distribution of phenotype tells us that there's 2 recessive alleles expressed by B6N mice that are controlling this pathway. They're controlling this CLIP phenotype. And we provisionally name them as class 2 presentation modifier A and B. And so I'm not going to tell you today what they are. I'll tell you that the B6J and N genomes have been sequenced, and using mini MUGA, and we were able to use the 51 coding variants that were known to start to map these genes. And then by creating additional SNPs and moving through the data, the mapping is nearly complete. We have 2 candidate genes that we think are likely the right genes. We aren't sure, again, so I'm not going to jump the gun and tell you what they are. I just will tell you that they are not, they have no known role in the pathway, and they're very surprising. So hopefully the main take-home message, on which is not really the best message ever, but it's that this pathway, although really well studied and thought to be completely understood, is clearly not understood. So I'm almost done here. I just have one last set of quick data that I'm going to present. So I've told you this story about immunity, and we really think that DO is playing an essential role in modulating the immune response. And remember, go back to the normal situation. This is an inhibitor, so it seems to be dampening immune responses. And this really comes back to this autoimmunity and this idea that we have the pathway is not completely understood. But this really begs the question, what about autoimmunity? Is DO really there then to modulate the peptide repertoire, spread it out, get a broader repertoire out there as a tolerance mechanism to prevent autoimmunity? And I'm going to go through this data really quickly. It was work done by Tatiana Glavinka and her husband, Sasha Chervansky at the University of Chicago. We contributed to this, but I think, and I put this in here, even though I played a small role in this, because it's really important. And what they did was they created 3 mouse models of autoimmunity in which they knocked H2o out. And the idea would be, if it's there for tolerance, if you knock it out, the mice should get severe autoimmunity or potentially get severe autoimmunity. So the first model they used were Dodm mice. So this is a model for type 1 diabetes. The mice spontaneously get diabetes. And when they followed the knockouts compared to the wild types over time, there's absolutely no difference. So at least in type 1 diabetes, H2o knockout doesn't seem to make it worse. It doesn't exasperate it. It doesn't make it better. It doesn't make it worse. It seems to have really very little impact. So they also tested it in a model for lupus. This is the B6NZM model, if anybody cares. And this is published. I'm going to go through this pretty quickly. The take-home message is the same. There's really no difference in the parameters that are measured for looking at lupus in this mouse model. So ANA, anti-nuclear antibodies. There's no difference between wild type and knockout mice. No difference in complement scores, IgG scores, and the GN scores. So really, again, in another model, you knock DO out and the mice are not getting spontaneous autoimmunity. This is the part of this we contribute to a little bit, and we then switched to an induced model of autoimmunity, a model of multiple sclerosis, where you inject mice with a peptide from myelin basic protein together with pertussis toxin, and you induce this EAE disease in mice. And what you can see in 3 different cohorts of mice, so this is mice in Chicago, and then these were mice in New Jersey in my colony. And in all 3 situations, we saw really no significant differences in disease development. So this kind of brings back this idea, is H2O involved in autoimmunity? And in these 3 mouse models, clearly not. So is there a role in human immunity? I leave this, I really think the verdict is still out, because there's some differences between the proteins in mice and humans that make me think that maybe in humans there really still could be a role. And we can talk about that in the question section if anybody's interested. So what we are really thinking is that maybe it's not autoimmunity, and it's really all about immunity. And we continue to work on these infections that we've already worked on. We're moving out into other pathogens. And then the main thrust of really what my lab is going after right now are these other modifiers of the pathway. So I just want to, this is crazy. I mean, I've spent a lot of time thinking about, could there really be more to this pathway? And I started reading about evolution just because I did for another reason. And actually I discovered that it's pretty interesting. So it turns out that MHC and T cells developed very early in evolution. So the sharks and the bony fish had T cells and MHC molecules, but no DM. So this pathway was around a long time, millions of years without DM, and was functional. And it's only in the amphibians that you first see DM being expressed, or being homologs being present. So this is going to modify the pathway. And then interestingly, DO is actually a relatively late adaptation where emergence, where you see it only in warm-blooded mammals. So it first appears in the birds. So this really kind of had me thinking back about pathways that, for a long time, it really functioned without these other modulators of the pathway. And, peptides were clearly getting loaded because you have T cells back here. And then you have these modulators of the pathway that we think we're all about peptide loading, but maybe they're doing other things in this pathway that we have yet to discover. And so this is my last slide. I just want to remind you that, the mass spec really does show that there's a change in the peptide repertoire. What this means on the immune response is really what I'm most interested in. And we really think it's much more complex and much more exciting than was originally thought. So an old pathway that may have new players and new important aspects to it that I hope to come back in a couple of years and be able to tell you about. And then no work is complete without a huge acknowledgement to the people that do the work. I actually still work in the lab, so I have a small lab. I need to acknowledge the past people who did a lot of the work I talked about. Austin Graves was a graduate student in my lab that did all the DOA work. Francesca was involved in all of the OB work and the DOB mutagenesis studies with help from lots of others along the way. Julie Dobkin is taking on the NIH mice and is working on this in my lab now as a graduate student. Lou is a technician, a senior technician who without we could do nothing. He can do everything with mice that you could ever imagine. Sarth was a technician in the lab who just left for med school, and I have to say the med school and the patients that ultimately get him are going to be very lucky because he's a very talented individual. We have Linda Warda, who's an undergrad who helps us with everything. A lot of what I do at Rutgers is in collaboration with Dr. Derek Santangelo's lab and the people in his lab, including Jeff Vieth, and then Tatiana in a group at the University of Chicago. And Tatiana's lab is also small. She also does a lot of the studies that I showed you that we work together in the lab. I often go to Chicago and we spend weeks in the lab together. It's fascinating and so much fun. And then her graduate student, Emily Collin, who's now gone back to med school. So she's an MD PhD student. And then Sasha and Ali, and then with some help from our colleagues in Berlin who've worked on some of this with us. And with that, I'm happy to take questions, and I thank you for listening.